exercise. The respiratory pump also helps propel blood through the thoracic and abdominal cavity veins, driven by rhythmic changes in pressure in the thoracic and abdominal pelvic cavities that occur when you breathe. During inspiration, high pressure in the abdominal pelvic cavity creates a pressure gradient that pushes blood in the abdominal veins upward, and low pressure in the thoracic cavity allows the thoracic veins to expand. When you blow out, the abdominal veins expand and fill with blood, while the thoracic veins are squeezed. And so that rhythm goes on as you breathe. Now, occasionally what can happen is that the blood can pool in the lower extremities and result in what we call varicose veins. This is characterized by dilated, bulging, hardened veins, generally in the lower limb, where the blood tends to pool, especially if you have an occupation that involves a lot of standing. The condition that decreases the rate of venous return is essentially this blood pooling in the lower extremities. It can be exacerbated by pregnancy, standing upright for prolonged periods, and obesity, and can cause blood to pool in the veins of the lower limbs. It also has a genetic predisposition. The lower limb superficial veins are not supported by skeletal muscle pumps, so extra blood volume stretches them and causes the enlarged regions to become visible beneath the skin. We're talking here about the superficial veins. High pressure in the abdominal pelvic cavity during defecation or childbirth decreases the return of venous blood from the anal veins, also superficial and not well supported by surrounding tissue, and this can result in a condition known as hemorrhoids, which can be dangerous because of their location. If they bleed, they can become infected, and you can end up with a condition known as sepsis. Okay, that brings us to the end of the first half of this chapter. I will see everybody in the second half of Chapter 18. Okay, our next topic is the maintenance of blood pressure. Mean arterial pressure has to be constantly maintained around 95 millimeters of mercury. Any deviation from this set point is going to trigger mechanisms from the nervous, endocrine, and urinary system to restore the blood pressure to its previous level. Short-term control of BP is primarily accomplished by nervous system and hormones of the endocrine system. Such short-term effects are generally achieved by adjustment of resistance and cardiac output. Nervous system maintenance of blood pressure involves the main branch of the nervous system, the autonomic nervous system, and its two divisions, the parasympathetic and the sympathetic. Sympathetic axons release norepinephrine and epinephrine onto cardiac muscle cells and smooth muscle cells of the blood vessels and generate two immediate changes, both increasing blood pressure. The increase in heart rate and contractility, which increases cardiac output, and vasoconstriction of all types of vessels, but especially arterioles, which increases peripheral resistance. In the parasympathetic division of the ANS, the vagus nerve releases acetylcholine into certain cardiac pacemaker cells, particularly the SA and AV node, as well as the atrial cardiac muscle cells, decreasing blood pressure by slowing heart rate and decreasing cardiac output. In the baroreceptor reflex, negative feedback responds to an increase in blood pressure. Opposite feedback loop will occur if the systemic blood pressure decreases and the receptors detect less stretch, and the rate of depolarization of the baroreceptors decreases. In response, the medulla stimulates the sympathetic nervous system to increase blood pressure. Okay, so you can see how short-term maintenance is going to operate here. The Valsalva maneuver is used to test the baroreceptor reflex. The subject bears down and tries to expire against a closed glottis. This occurs in coughing, sneezing, defecation, and heavy lifting. It raises the pressure in the thoracic cavity and reduces the return of venous blood to the heart and causes a drop in blood pressure. This should trigger the baroreceptor reflex and generate an increase in heart rate. The effects of chemoreceptor stimulation include the detection of oxygen and carbon dioxide and pH levels in the blood. This plays a key role in the regulation of breathing, but also affects blood pressure. The receptors respond mostly to oxygen levels in the blood. In central chemoreceptors, we respond to a decrease of pH in the interstitial fluid in the brain. This triggers another feedback loop that indirectly increases the activity of the sympathetic neurons and results in vasoconstriction and a rise in blood pressure. Now, you might wonder why it is that pH has such a profound role in regulating blood pressure. Well, what happens when carbon dioxide builds up in the blood is it produces carbonic acid, which reduces the pH of the blood. So as the pH drops, it's incumbent on the body trying to eliminate more CO2 by increasing both the heart rate and the respiratory rate. And so the central chemoreceptors are going to have a dual-pronged effect. They're going to pick up the change in pH as the carbon dioxide diffuses into the interstitial fluid in the brainstem. That's going to cause the pH of the cerebrospinal fluid to drop. The central chemoreceptors will then kick in and increase the heart rate and the respiratory rate. The peripheral chemoreceptors are going to respond to oxygen, CO2, and pH levels, but primarily oxygen levels. The endocrine system works with the nervous system to maintain nearly all aspects of homeostasis, including blood pressure, although hormonal responses are much slower. Following highlights the endocrine system's short-term regulation of BP. Hormones that control cardiac output include epinephrine, norepinephrine, and T3 and T4. Hormones that control resistance include epinephrine and norepinephrine from the adrenal medulla, which promote vasoconstriction, thus increasing peripheral resistance and elevating blood pressure, and angiotensin II, which is a potent vasoconstrictor. ANP, which comes from the atria, causes a decrease in peripheral resistance as a result of producing a decrease in blood volume by eliminating more sodium in the filtrate, which causes an increase in urine production and a decrease in blood volume and blood pressure. Long-term maintenance of blood pressure falls to the urinary system and certain hormones of the endocrine system that hit the kidneys and control BP by increasing or decreasing the amount of body water we lose in the urine. The endocrine system regulates blood volume through the release of ANP and angiotensin II, as well as ADH and aldosterone. When blood pressure increases, atrial cells kick out ANP, and this causes the kidneys to excrete more water as it follows the sodium into the filtrate out of the body. This decreases blood volume and drops blood pressure. When blood pressure decreases, ADH secretion triggers thirst and increases the amount of water retained by the kidneys, thus raising blood volume and blood pressure. Now, renin is secreted from the kidneys, and it's triggered when blood pressure drops. It begins the process that activates angiotensin II, induces thirst, causes sodium ion retention, and as a result increases blood volume. Angiotensin II also triggers the release of aldosterone from the adrenal cortex, causing the retention of sodium and water from the kidneys, and this also increases blood volume. The urinary system's control over blood volume includes if rising blood pressure is indicated, more water will flow through the tubules of the kidneys than those cells can return to the blood. The water is lost from the body as urine. The blood volume and blood pressure go down. If the blood pressure decreases, less water flows through the kidney tubules, and these cells have more time to reclaim water and return it to the blood, and this results in decreased urine production and an increase in blood pressure and volume. The maintenance of blood pressure by the nervous, the endocrine, and urinary systems is summarized here, and each panel indicates the effects the systems have on each factor that impacts blood pressure, blood volume, peripheral resistance, and cardiac output. Some disorders of blood pressure include hypertension and hypotension. These are deviations of blood pressure outside homeostatic norms. In hypertension, we don't really know the cause, while in secondary hypertension, we can definitely assign a cause. Basically, this is going to be elevated blood pressure. In the case of hypertension, we're talking about a blood pressure higher than 140/90. Hypotension is defined as a systolic pressure lower than 90 and a diastolic lower than 60 is generally diagnosed as such only if the individual shows symptoms. Severe hypotension, known as circulatory shock, can lead to loss of consciousness and organ failure. Blood pressure is insufficient to deliver oxygen and nutrients to the cells, which can then be fatal. The most common cause of hypotension is reduced blood volume, which can occur as a result of blood loss, fluid losses from diarrhea, vomiting, overuse of diuretics, or insufficient fluid intake. Severe blood loss can lead to hypovolemic shock, which is fatal unless we restore blood volume. A decrease in heart rate can result in hypotension. This is generally due to medications prescribed to treat hypertension. A decrease in stroke volume is most commonly caused by heart failure, which is the inability of the heart to effectively pump. In severe heart failure, we can produce dramatic drops in cardiac output that can result in cardiogenic shock. Excessive vasodilation can produce profound hypotension as well. Many conditions can cause excessive vasodilation, including medications for hypertension, abnormalities in autonomic function, and a decrease in blood pH. Another potential cause of excessive vasodilation is the release of histamine into the blood following a severe allergic reaction known as anaphylactic shock. Excessive vasodilation occurs with certain bacterial infections, and we term this septic shock. So let's talk a little bit about the actual exchange of oxygen, nutrients, and waste products at the tissues. This is what takes place at the capillary beds. Capillaries are generally found in clusters called beds that wind their way between the tissues of the body. Blood flow to a tissue through a capillary bed is known as perfusion. It's tightly regulated to ensure that metabolic needs of tissues are met at all times. The capillaries are very thin vessels with walls that are only about 2/100ths of a micron thick. Each capillary consists of an endothelia rolled into a tube and a small amount of basal lamina secreted by the endothelial cells. Structural and functional features include parasites, which are cells found around some capillaries that have contractile filaments that appear to control blood flow through the capillaries. Capillaries average about 50 microns in length and 8 to 10 microns wide. The walls of most capillaries consist of 1 to 3 endothelial cells joined by tight junctions. They curl around the capillary's entire circumference. Capillary exchange describes the movement of nutrients, gases, ions, and waste that have to cross the wall and travel between the blood and the capillary and tissue cells. The movement of materials is known as capillary exchange. Materials are generally exchanged through 3 mechanisms, including 2 types of diffusion and transcytosis. If we look at diffusion and osmosis, this occurs through gaps and pores. Some capillaries have tiny pores within their endothelial cells known as fenestrations. The result is that water as well as small substances like monosaccharides and amino acids that are dissolved in water are able to move freely through these pores as long as there's a gradient driving them. If we look at the movement of materials through the membranes of endothelial cells, we know that lipid-soluble material such as oxygen, CO2, and other compounds such as steroid hormones or lipophilic drugs can generally enter and exit the capillary by diffusing across the membrane from one side of the endothelial cell and out the other side. The substances are then going to enter either the interstitial fluid or into the blood. In transcytosis, large molecules cross the endothelial cells. The molecules are taken into the cell on one side via endocytosis and ejected on the other side via exocytosis. Again, this is a form of active transport. Both diffusion and transcytosis involve a substance crossing the entire width of the endothelial cell, and this is another example of the structure-function core principle. The types of capillaries are shown here. They're found in different parts of the body, and they have slightly different functions, and as a result, slightly different structures. They include continuous, fenestrated, and sinusoidal capillaries. The majority of capillaries in the body are continuous capillaries found in muscles, skin, nervous, and connective tissue. Continuous capillaries are the least leaky. They permit the fewest substances to enter or exit the body by extracellular routes because their endothelial cells are held together by tight junctions. Fenestrated capillaries are much leakier because they contain fenestrations. Those are the pores that let materials go through. The pores allow diffusion to take place more quickly than in continuous capillaries. The fenestrateds are therefore located in places that require substances to rapidly enter or exit the blood. Sinusoidal capillaries are the leakiest. The name is often simply shortened to sinusoids. They have a discontinuous sheet of endothelia, an irregular basal lamina, and large pores in their endothelial cells. They're typically 3 to 4 times larger in diameter than other capillaries and have an irregular shape because their boundaries are determined by the organ they sit in. They're located in organs and tissues such as the liver, the spleen, and lymphoid organs, as well as the bone marrow, and facilitate the transfer of large substances such as blood cells and large proteins between the interstitial fluid and the blood. The flow of blood that takes place within the body's capillary beds is collectively called microcirculation. It involves two types of vessels: true capillaries where materials are exchanged, and a small central vessel as shown here. True capillaries form an interweaving network with multiple anastomoses fed by the proximal end of a central vessel, which is formed by either a small terminal arteriole or meta-arteriole. Each capillary meta-arteriole junction contains a precapillary sphincter that controls the amount of blood flowing into the capillary. At rest, only about 25% of the body's capillary beds are fully open. This helps maintain blood pressure. If all the capillaries were open at the same time, a drop in blood pressure would cause you to lose consciousness. True capillaries are drained at the distal end of the capillary bed by a portion of the central canal called a thoroughfare channel. These channels lack precapillary sphincters, which reflects the fact that this is drainage at the end of the capillary bed. When the precapillary sphincters close, blood flows into a straight meta-arteriole through a thoroughfare channel and into a venule without passing through the capillaries. The blood does not flow backward into the true capillaries from the thoroughfare channels because the pressure gradient favors the movement of blood in one direction only, again as shown here. Okay, and so you could almost think of this as draw. Okay, as the blood flows rapidly through this thoroughfare channel, the draw is going to tend to pull the blood directly into the venule. Tissue perfusion is regulated by local factors within each individual tissue. This is called autoregulation. This ensures that the correct amount of blood is delivered to match a tissue's level of activity and maintain two types of autoregulatory controls: myogenic and metabolic control. The myogenic mechanism counters a change in blood flow by altering arterial resistance. The velocity of blood flow is related inversely to the resistance, so if the resistance increases, the velocity decreases. The mechanism slows blood flow by increasing resistance when arterial pressure rises. The mechanism speeds up blood flow by decreasing resistance when the arterial pressure drops. Both changes maintain local tissue perfusion at a constant level and ensure that gases and substances continue to be exchanged in adequate amounts in the face of fluctuating blood pressure. Metabolic controls are mediated by chemicals present in the interstitial fluid surrounding the capillaries. Chemicals are the result of metabolic activities, those that produce ATP, the cell's form of usable energy. In cells that are producing ATP rapidly, low concentrations of oxygen and high concentrations of carbon dioxide and hydrogen cause smooth muscle cells of the local arterioles to relax. This dilates the arterioles and increases perfusion, ensuring adequate oxygenation and nutrient delivery to actively metabolizing cells. In cells that are producing ATP slowly, high concentrations of oxygen and low concentrations of carbon dioxide and hydrogen cause constriction of local arterioles and a decrease in tissue perfusion. Now, this is a very important point. We should point out that in systemic tissue, this is absolutely true. Okay, so we'll do systemic tissue here in black. So in systemic tissue, if we see an increase in CO2, a decrease in pH, or a decrease in oxygen concentration, we're going to see, this is going to lead to an increase in perfusion. Okay, so this is in systemic tissue. In the lungs, that should be a C there, systemic. In the lungs, it's a different story. Okay, we'll talk about this when we get to the chapter on the lungs, but I just want to point out while we're here, in the lungs, if we see in the tissues an increase in CO2, a decrease in pH, or a decrease in oxygen, we're going to see a decrease in perfusion. You might wonder, well, why would that be? It's a phenomenon called ventilation-perfusion coupling that only perfuses regions of the lungs that are well oxygenated. Those that are well ventilated are going to get the bulk of the blood, and those that are the most poorly ventilated are going to receive less of the blood. Okay? Gravity also plays a role with this, but this ventilation perfusion coupling is critical in order to make the best use of the lung tissue that we have. Okay, so we should write up here that this is in pulmonary tissue, lung tissue. Each organ has its own unique requirements in terms of blood flow and nutrients. The heart gets about 5% of the total cardiac output through the coronary circulation. This perfusion pattern through the coronary circulation is the opposite of the rest of the systemic circuit. Heart tissue perfusion decreases during systole. The main local autoregulatory mechanism of the cardiac muscle tissue appears to be metabolic control, in particular the concentration of oxygen in the cardiac interstitium. Low interstitial fluid oxygen triggers the production of vasodilators that dilate the arterioles that serve the myocardium and increase perfusion. Now you might wonder, well, why is it that heart tissue perfusion decreases during systole? Well, during systole, when the ventricles are squeezing, what's happening is that the blood is being ejected into the aorta and towards the body. In diastole, when the ventricles relax, the blood's bouncing back off of those aortic semilunar valves. It then fills the coronary arteries. Well, I won't say it fills it. It increases the amount of blood flowing into the coronary arteries, and that increases the tissue perfusion for the heart itself. The brain is extremely intolerant of ischemia. A sudden decrease in tissue perfusion in the brain results in loss of consciousness in seconds. The brain accounts for 2% of the total body mass but gets 15% of the cardiac output. Autoregulatory mechanisms, including myogenic and metabolic control, maintain cerebral blood flow at a constant rate of about 750 milliliters per minute. Despite the constant blood supply, the blood flow within the brain to different areas varies with neuronal activity. In part due to standard metabolic controls, areas of the brain that have higher activity get more oxygen and produce more CO2. Certain neurotransmitters are direct vasodilators, and others act on astrocytes that are in contact with the cerebral arterioles. When stimulated, the astrocytes induce production of vasodilators. This opens arterioles and increases the perfusion to active regions of the brain. Now keep in mind, when we're talking about the brain, the blood never comes into direct contact with the neural tissue. Okay? However, blood vessels are found throughout the brain, the brainstem, and the spinal cord. So how is it that the oxygen and nutrients get there? It's via the astrocytes. They are the mediators in the CNS. Okay. In the peripheral nervous system, remember that we have satellite cells and Schwann cells that mediate a lot of those processes. Okay. Again, the formed elements of blood never come into direct contact with nervous tissue. In skeletal muscle, blood flow changes dramatically during exercise, increasing as much as 50-fold. This is known as hyperemia, which is a mechanism that lies in the structure of the arteries that supply skeletal muscle. An artery that enters a skeletal muscle is a feed artery, which branches into multiple arterioles and ends in terminal arterioles that supply a capillary bed. During resting conditions, resistance in the feed arteries is high, and many of the terminal arterioles are constricted, and this limits perfusion. But when you exercise, metabolic conditions around skeletal muscle dilate the arterioles. This produces an increase in tissue perfusion as more capillary beds open, but not enough of an increase to meet the demands of vigorously working skeletal muscle. As the exercise continues, vessels proximal to the terminal arterioles also dilate and dramatically increase perfusion. This progressive stepwise vasodilation allows skeletal muscles to precisely match perfusion to the degree of exercise. Your skin is the largest organ in your body. The blood supply is located in the dermis. Oxygen and nutrients have to diffuse from the capillaries in the dermis to the epidermis. Local autoregulation of the skin's blood flow does take place in response to temperature. Placing something warm directly on the skin produces vasodilation, whereas a cold object will produce vasoconstriction. Most important in the skin's blood flow is sympathetic nervous stimulation. Neurons regulate blood flow to the skin as part of thermoregulation. The sympathetic nervous system alters skin perfusion through vasoconstriction in order to conserve heat or vasodilation in order to release it. Okay, let's talk a little bit about how pressure produces flow but also promotes water movement. Hydrostatic pressure is defined as the force that a fluid exerts on its container walls. It's inversely proportional to volume. For a fixed volume of fluid, the smaller the container, the higher the pressure, and vice versa. Movement of water across a capillary is driven by filtration. Movement of a fluid by a force such as pressure or gravity is what drives this. Pressures that are at work at the capillary include two basic pressures that drive water movement: hydrostatic and osmotic pressure. These promote movement in opposite directions. Hydrostatic pressure drives water out of the capillary, while osmotic pressure draws fluid in. Blood is fluid in the vessel that is creating hydrostatic pressure. As a result, blood pressure is equal to hydrostatic pressure. Blood flows through an area of high hydrostatic pressure to one of low hydrostatic pressure through the capillaries until the gradient disappears. This is a passive process known as filtration. So you can see here filtration in action under hydrostatic pressure. Hydrostatic pressure in a capillary changes from its arterial end to its venular end, from 35 millimeters of mercury down to 15. Hydrostatic pressure of interstitial fluid is so low that it is functionally zero. The result is that the resistance from the interstitial fluid, the pressure in the opposite direction, is minimal. As a result, there are steep hydrostatic pressure gradients on both ends of the capillary that drive water out of the capillary and into the tissue fluid. Osmotic pressure involves the movement of water from an area of high to low water concentration. The particles that determine this dictate the solution's osmolarity. A solute particle in a solution will exert a force or a pull on water molecules that's called osmotic pressure. The osmotic pressure is determined almost exclusively by the number of particles and not their size. The osmotic pressure in capillary beds is about 25 millimeters of mercury and is created exclusively by large proteins in the blood, particularly albumin. Proteins too large to leave the capillary remain inside. As a result, the osmotic pressure remains consistent through the capillary's length, and osmotic pressure is very low in the interstitial fluid. The difference in osmotic pressure creates an osmotic pressure gradient known as colloid osmotic pressure, or COP. Colloid osmotic pressure draws water into the capillary through osmosis. The terminology for osmotic pressure may sound confusing because we tend to think of a pressure as something that pushes, but as we've mentioned, osmotic pressure doesn't push, it pulls. To understand why it's called pressure, first know that a pressure is simply a force applied to a molecule or a group of molecules. Second, understand that a force is defined as a push or a pull. Think of it this way. You have a wagon at the top of a hill and you apply a force and push it down the hill. To get the wagon back up to the top of the hill, you apply another force and pull it up the hill. In both cases, you're applying a force that moves the wagon. In this analogy, the hydrostatic pressure is the force that pushed the wagon at the bottom of the hill, and the osmotic pressure is the force that pulled it back up. The net filtration pressure in the capillary is a combined force of the colloid osmotic pressure and the hydrostatic pressure gradient that drives water in opposite directions. Hydrostatic pressure pushes water out of the capillary bed, and colloid osmotic pressure tends to pull it in the opposite direction. The difference between these two gradients is the net filtration pressure. At the capillary's arterial end, the hydrostatic pressure is 35 millimeters of mercury, and the colloid osmotic pressure is 22. Hydrostatic pressure is defined as the force that the fluid exerts on the walls of the container, the blood vessel. The colloid osmotic pressure is the osmotic pressure of the dissolved substances in the blood plasma. If we subtract the COP from the hydrostatic pressure, we get the net filtration pressure, which works out to 13 millimeters of mercury out of the capillary. This force drives water out of the capillary by filtration because the hydrostatic pressure is greater at that end. At the capillary's venular end, the hydrostatic pressure is 15 millimeters of mercury, while the colloid osmotic pressure remains unchanged at 22. The hydrostatic pressure minus the colloid osmotic pressure at this end results in a net filtration pressure of negative 7 millimeters of mercury. This negative number means that water flows into the capillary at the venous end of the capillary bed. This is absorption. As a result, water is absorbed into the capillary by osmosis. Now, does all the water get recovered? No. About 15% of the fluid that leaves at the arterial end of the capillary bed enters the lymphatic system and is then filtered by the lymph nodes and returned to the venous return to the heart near the right and left subclavian veins, with a right thoracic duct, or where the right lymphatic duct and the thoracic duct allow the fluid, the lymph, to re-enter the circulation. The same equation can be used to determine the overall net filtration of the whole capillary. The overall hydrostatic pressure is 13 millimeters of mercury. This pushes the water out by filtration. The overall colloid osmotic pressure is 7 millimeters of mercury, pulling the water in by absorption. The net effect, if you subtract these two values, is 6 millimeters of mercury. The overall net filtration pressure favors filtration of water out of the capillary. Edema is a condition characterized by an excessive amount of water in the interstitial fluid. Common causes of edema include an increase in capillary hydrostatic pressure gradients due to hypertension, or a decrease in colloid osmotic pressure due to liver disease, cancer, starvation, or other disorders. Peripheral edema is the pronounced edema found in the hands and the feet, where hydrostatic pressure gradients are already slightly higher because of gravity. It's even more profound if it results from decreases in colloid osmotic pressure, where excess water tends to accumulate in the interstitial fluid of the abdomen, a condition known as ascites. And you might wonder, okay, well, how does that happen? An example would be an individual who's got a protein-poor diet, or somebody in liver failure. The result of both of those conditions can be a lack of albumin. The lack of albumin means that the colloid osmotic pressure pulling fluid back into the circulatory system is reduced. The result is that the fluid tends to leave the blood and enter the surrounding tissues. And that's why you see on those spots that they run for third world countries needing nutrition, the distended abdomens of the children that have the protein-poor diet, because they can't make enough albumin for their blood to hold on to its fluid volume. You also see this in individuals that are in uncontrolled cirrhosis, as the liver fails to make the critical blood protein albumin. Okay, I appreciate everybody listening. I'll join all you in the next podcast. Remember to review this material, as it will be covered on the exams and the PRS quizzes. Thank you very much.